So let's talk now about eukaryotic cells. In this segment, we are going to address both the structural segment of eukaryotic cell, but also the functional segment of the components that make up eukaryotic cells. Now, we said that eukaryotic cells are much more complex than prokaryotic cells. We said that they were larger. Now, in terms of complexity, we said that in addition to the nucleus that is going to house the genetic information, you are going to have many compartments. And these compartments are delimited by membranes. In other words, membranes set the limit from one compartment to another compartment. And let me say that the advantage for eukaryotic cells to develop these different membrane-bound organelles is to isolate different metabolic processes. By isolating different metabolic processes, this optimizes the functioning of the cell. This is a very important segment to keep in mind as to why the cells evolved to develop membrane-bound organelles. Now, what do we call an organelle anyway? An organelle, it's a structural part of the cell that has a specific function, that is able to carry a specific function that another part of the cell is not carrying. Okay, so here we have side by side a model of animal cell and a model of a plant cell. From this slide, really, we can go through all the different components of an animal cell and the different components of a plant cell that we are going to address a little later. For instance, we can see that we have the nucleus, right? The nucleus is made up of what? You have the nuclear envelope, right? You're going to have the nucleolus. Now, not mentioned here, you're going to have the genetic information, so DNA, that is organized in the form of chromosomes, right? And these chromosomes, some of them are loose, some of them are compact, but we're not going to dive into that. We have the we have the plasma membrane, okay? The centrioles that are located here. What are the centrioles and what role do they play in the cell? We are going to see that. The mitochondria. We're going to have the cytoskeleton. Among the cytoskeleton, we're going to have the actin filaments, the microtubules, and the intermediate filaments. These are the three types of protein fibers that make up the cytoskeleton. We have, obviously, the cytoplasm, which is actually the semi-fluid substance in which all the organelles are going to bath, right? We have the endoplasmic reticulum, including the rough endoplasmic reticulum that has ribosomes attached to them, okay? And the smooth endoplasmic reticulum, okay? They don't all have the same function. We have the ribosomes, which function is going to be to do protein synthesis. We have the Golgi apparatus. We have the peroxisomes. We have the lysosomes. Now, a lot of the structure that we find in animal cells, we're going to find in plant cells. However, we are going to have some major differences, including, for instance, the presence of chloroplast in plant cells that you don't find in animal cells. What do chloroplasts do? We are going to address that. We're going to have the cell wall that you find in plant cells that you don't find in animal cells. Very important to mention that. Otherwise, most of everything else that is present in animal cell, you're going to find them in plant cell and vice versa. Okay? All right. So the organelles in eukaryotic cells allow the cells to be organized in many compartments. These compartments are going to isolate functions, as I already mentioned earlier. Now, to go from one compartment to another compartment, you're going to have the exchange of material that is going to be controlled by the development of what we call transport vesicles. These transport vesicles are going to, from one compartment that we're going to call compartment A, a vesicle is going to bud off from compartment A. As this vesicle bud off, it's going to leave with a cargo. And that cargo, in this particular instance, it's proteins, right? These proteins, they are going to leave this compartment A, migrate, and find themselves in compartment B. That is actually another organelle. So we have organelle A and organelle B. And notice that when you look at the inside of organelle A, it's not the same color as organelle B. This is just to tell you that the activities that are happening in organelle A do not happen in organelle B. And that's how those two are going to be different, right? We see that in the case of protein production, for instance, some proteins that are designed to go outside of the cell, when the translation of this protein starts, once the ribosomes start producing the proteins, these proteins are going to migrate into the rough endoplasmic reticulum. In the rough endoplasmic reticulum, the proteins there are going to receive some modification. After this modification are completed, then the proteins are going to end up in a vesicle that is going to bud off, as you see here, okay, from that rough endoplasmic reticulum. And from there, the vesicle is going to travel until it reaches a compartment that we call the Golgi apparatus. In the Golgi apparatus, the protein is going to be released in here, and in there, the protein is going to undergo further modification. These modifications are going to determine the fate of the proteins, right? The proteins that are intended to go outside of the cell will receive a particular types of modification that are such that when the system sees this modification, the system knows that, well, this protein belongs to outside of the cell, right? So here we have a micrograph that shows you the cell membrane, here, the nucleus, here, a whole bunch of organelles here that are membrane bound, right? The mitochondria, actually these are the mitochondria that we see here. You see there are many of them. What else can I show you? You have a whole bunch of vesicles, right? A whole bunch of vesicles. So this is a micrograph taken with an electron microscope. And how do we know this? We know this because we can see the details inside of the cell. If it was a scanning electron microscope, we would see the surface of the cell. All right. So this table, it's really here to actually give you a quick snapshot of the different components that make up eukaryotic cell in terms of structure and the characteristics of each component. Now, we are not going to go through the slide. However, I'm putting the slide here for you to use it as a guide. Okay? If I was to focus on few things in relation to eukaryotic cell structure, what should I focus on? Right? The plasma membrane. For instance, know that the plasma membrane is actually a phospholipid bilayer, right? But that phospholipid bilayer, the lipids are not equally organized and equally modified, which allows us to say that the plasma membrane is asymmetric. Now, here is the thing. I will not bug you with that. But what I will bug you with and what I expect you to know is that this plasma membrane is semi-permeable, just like we saw with the plasma membrane of bacteria. The plasma membrane is going to contain, in addition to phospholipids, we're going to have some transport proteins, right? You're going to find some receptors. You're going to find some proteins that are essential in cell communication, in cell-to-cell connection, so tissues can develop, and many other functions that I'm not going to dive into. So take the time to go through this slide. This can be very helpful for you just to have a snapshot. And this is some more in terms of comparing prokaryotic cells and eukaryotic cells, right? A really nice snapshot that shows you what are the very specific components that you need to remember in terms of characteristic, in terms of similarities and differences. Please review the slide. All right. So, the plasma membrane of eukaryotic cells. Between eukaryotic cells and prokaryotic cells, the phospholipids have the same basic structure. However, the phospholipids are going to be modified in different manner. I'm not going to bug you with the details. I told you that I'm not going to bug you with that. However, the phospholipids, they are mingled with proteins, right? Just like we saw earlier with prokaryotic cells. Then these proteins, many of them are going to serve as receptors. Remember that. Animal cells, for instance, live in a very complex system. You think about your body. You have there blood cells, you have liver cells, you have skin cells, you have kidney cells, you have cells of the digestive system, you have cells of the circulatory system, you name it. You have different kind of cells. Cells have to communicate with each other. They have to be able to recognize each other. They have to know in which context they are, right? So they need these receptors to feel and eventually to develop a response, right? So, membranes of eukaryotic cells, and particularly animal cells, they are going to have cholesterol. Cholesterol is essential to maintain the fluidity of the plasma membrane. When it's cold, the cell is going to need a lot of cholesterol in the plasma membrane so the membranes do not freeze. When it's warm, the cell is going to have less cholesterol in the plasma membrane so the plasma membrane does not melt away. So cholesterol plays a very important role in the structure of the plasma membrane in animal cells. In fungi, in place of cholesterol, you have another sterol, it's ergosterol, okay, that plays a similar role. Now, the plasma membrane of eukaryotic cells, we are going to have what we call lipid rafts. These lipid rafts allows the cell to recognize the context in which they are, okay, to respond to signals so that they can behave appropriately, okay? The membranes are also going to play a very important role in eukaryotic cells in what we call electrochemical gradient. However, in animal cells, this electrochemical gradient does not happen in the plasma membrane. It does happen in the membrane of mitochondria, and specifically the inner membrane of mitochondria. That's in animal cells. In plant cells, however, this happens both in the inner membrane of mitochondria and in inner membranes of chloroplast. And the chloroplasts are going to contain some little structures that we call thylakoids. These thylakoids, in the membrane of thylakoids, this is where we're going to find the machinery, the electron transport chain, that is going to be responsible for the development of the electrochemical gradient. All right? So, let me see. The last thing I want to mention is that the plasma membrane of plant cells and animal cells is not involved in ATP synthesis, as opposition to what we saw in bacterial cells. Okay? In eukaryotic cells, mitochondria are responsible for ATP production, and chloroplasts in plants are responsible for ATP production. All right. So I did tell you that you have a lot of membranes, and actually a lot of membrane-bound organelles, and that there is a close communication between compartments in these membrane-bound organelles, right? So that takes me to another aspect that is very much present in eukaryotic cells, that is not present in prokaryotic cells, is the concept of endocytosis and exocytosis. Now, what is endocytosis? Endo means in. Cytosis is cell. So endocytosis is bringing into the cell. Exocytosis, it's taking outside of the cell. So now in prokaryotic cells, we say that the chemicals and the particles are coming in and out via proteins. Some of them are doing facilitated diffusion. Some of them are doing active transport. We mentioned the ion channels, right? And we also talked about aquaporin. However, here we have exocytosis and endocytosis. Now, how about exocytosis? Exocytosis, you have three kinds of exocytosis. One, it's when the cell needs to bring in or wants to bring in or is bringing in some hard, solid particles. For instance, your white blood cells can go after a bacterium that is infecting your body because you ate a Subway sandwich that was spoiled, right? The salmonella grew so much that that becomes a problem for your body. Now, when you bring that salmonella into your body has to fight back. And among the players that are fighting back, you have the white blood cells. And you have some white blood cells that are called neutrophils. Other white blood cells are called macrophages. Others are called dendritic cells. All the cells have something in common. It's that they can engulf bacteria by bringing them in. You have a vesicle that forms. That vesicle is called endosome. But before I talk about endosome, let me go back. So, whenever we have some bacteria, okay, actually, it's not here, I'm sorry. It's down here. Whenever we have some bacteria, the cell is actually going to engulf this bacteria or viruses and bring them in a structure that we call a phagosome. A phagosome. That phagosome right here is going to travel and merge with an inside component. That inside component that is found in the cell is filled with digestive enzymes. This digestive enzyme break down all kind of chemicals. It break down lipids, break down proteins, nucleic acids, carbohydrates, break down literally everything. So the phagosome is going to merge with this vesicle that we call the lysosome. Okay? So that we have one structure that we call a phagolysosome. That phagolysosome is going to give the opportunity for all these enzymes to break down the bacterium that we brought in, that the macrophage brought in, the neutrophil, or the dendritic cell. And after digestion, you're going to have a whole bunch of fragments of the cell, and these fragments are going to be ejected outside by way of exocytosis, right? So we have phagocytosis. Phagocytosis is also known as cellular eating, okay? Then we have pinocytosis. Pinocytosis means cellular drinking. This is when the cell takes up some droplets, okay? It can be proteins, it can be carbohydrates, can bring them inside of the cell by way of endocytosis and end up in a vesicle called endosome. That vesicle called endosome is going to travel and merge with a lysosome that again contains digestive enzyme. That lysosome is going to form with the endosome an endolysosome. The enzymes in that structure are going to cut down all the droplet that were brought, and after breaking all these parts down, they're going to be sent out by exocytosis if needed, obviously. And last but not least, you have a type of endocytosis that involves receptors. This is what we call receptor mediated endocytosis. In this receptor mediated endocytosis, the particles are going to attach on receptors, and then a vesicle is going to form. That vesicle is called endosome. That endosome is going to travel, merge with the lysosome, and again we're going to have the endolysosomes, and the content is going to be broken down, and the byproduct of that digestion is going to be dealt with accordingly. Okay, so endocytosis in animal system is going to involve three kinds of endocytosis. We have cellular drinking, that is pinocytosis, receptor-mediated endocytosis, and phagocytosis, that is cellular eating. Exocytosis, on the other hand, it's actually the reverse, of what we saw in endocytosis. In endocytosis, we brought some material into the cell. In exocytosis, we are bringing some material out of the cell. Now, that exocytosis does not only apply when the cell brings some material in by endocytosis. Say, for instance, your cell that we are going to call the lymphocyte B cells. These B cells, their job when they are mature and activated is to produce antibodies. These antibodies do not remain inside of the cell. They have to be released so they can travel into the blood and go target antigens, foreign particles, right? For this antibodies to end up outside of the cell, they have to be synthesized first, synthesized through the rough endoplasmic reticulum. They're going to travel to the Golgi apparatus, and after all modifications are done, these antibodies eventually are going to be sent outside of the cell by way of exocytosis. Okay? All right. So I told you about pinocytosis. We're not going to talk about it again. Receptor-mediated endocytosis and phagocytosis. And also, we talked about exocytosis, that is a reverse process of what we see with endocytosis. One can argue that, well, Dr. Jean, you talked about, some examples of endocytosis, pinocytosis, some examples of phagocytosis. What are some examples of receptor-mediated endocytosis? Well, you've heard of the low-density lipoproteins and the high-density lipoproteins, abbreviated LDL and HDL. Good cholesterol, high-density lipoproteins, bad cholesterol, low-density lipoproteins. The reason why I'm mentioning this is that the high-density lipoproteins, these are lipids that are wrapped by proteins. And the reason of this is because the lipids are hydrophobic and the cell contains a lot of water. So the lipids are the proteins are wrapping up lipids to form a huge particle. That huge particle is called high-density lipoprotein. These particles, the body does not want to see them freely floating into the bloodstream. They have to get away from the bloodstream. And how do they get away from the bloodstream, you have some receptors on the surface of our cells that are recognizing the HDL, high density lipoproteins, by way of receptors. And when there is a certain density of attachment of this lipoproteins on the receptors, there is going to be a vesicle that is going to form, and this is how the lipoproteins are going to come into the cells. Okay? All right. So how about protein synthesis? I did tell you that the ribosomes of eukaryotic cells are made up of two subunits, a large subunit that we called 60S, and a small subunit that we called 40S. Together, the ribosome is a 80S ribosome, which is different from what we saw in prokaryotic cells, where the ribosome, the whole ribosome, was 70S ribosome with a 50S large subunit and a 30S small subunit, right? These two ribosomes, they have the exact same function, which is to produce proteins, to do protein synthesis, right? However, they are slightly different in terms of structure. The proteins they are made up of. Now, let me digress. Ribosomes are a mixture of proteins and RNA, the ribosomal RNA. Now, the proteins that make up the ribosomes in eukaryotic cells are different from the protein that make up the ribosomes in prokaryotic cells. And this is how the system is able to tell them apart. Okay? Next up is the cytoskeleton. The cytoskeleton plays a tremendous role in the life of eukaryotic cells. Why? Because the cytoskeleton is essential for the cytoplasmic streaming. What this means is that when you consider a cell at a particular time in their life, organelles are organized in a certain way and at a specific location. What allows this organelle to sit at specific location at specific time? It's the organization of the cytoskeleton. Also, when you consider a protein that is being produced in the rough endoplasmic reticulum, how does the vesicle that is budding off from the rough endoplasmic reticulum know that the Golgi apparatus is this way? Well, the cytoskeleton is also the one that is going to create some kind of railroads. These railroads are going to make up tracks for this vesicle to travel, to go from point A to point B in a very specific direction, right? The movement is not random. Everything is tightly controlled. Bottom line, the cytoskeleton plays a very important role, okay? Now, in the cytoskeleton, we have the actin filaments. What is the role of actin filaments in the cytoskeleton? Movement. Think about, for instance, think about the contraction of your muscles. Your muscles wouldn't contract if it was not for the actin filaments. And if your muscles do not contract, you cannot move. I wouldn't do all the talking I'm doing right now. This is just an example as to the role of actin filaments in the body So actin filaments are also going to be essential and important for our cells to be able to crawl. I told you about the example of a white blood cells going after a bacterium. How do this white blood cell go after a bacteria? Do they have a flagellum? The answer is no. They don't have a flagellum. Okay? In fact, in human cells, in the human body, only one type of cell has a flagellum, is the sperm cells. All other cells don't have a flagellum. How do these cells move around? They are able to move around by crawling. They crawl around relying on actin filaments. Okay? Very important to remember. Our next step, it's the microtubules. The microtubules are going to be essential at organizing organelles in the cytoplasm, what we call cytoplasmic streaming. Okay? Now, microtubules are also essential for cell division. How so? Well, during cell division, the cell has to copy its genetic information and distribute its genetic information equally among daughter cells. Think about it. When you have a cut, your skin has to repair, right? When your skin repairs, you don't want your skin cells to be different one from another. Similarly, if you get a burn, right, your cells have to be replaced. And you don't want the cells to be different one from another, right? So they have to receive the exact same amount of genetic information. The microtubules are responsible for this. Okay? Microtubules are responsible for this. Finally, the intermediate filaments. The intermediate filaments are by far the less well understood cytoskeletal filaments. However, we know that this intermediate filament play a very important role at providing mechanical strength to the structure, whether we're talking about the nuclear envelope or we're talking about the whole cell itself, right? Okay, so we have the actin filaments, the microtubules, and the intermediate filaments. One can ask, well, Dr. Jean, what are all these little guys right here? Now, those little guys, in the actin filaments, these are actin proteins. So these are actin proteins. Okay? So these actin proteins are going to polymerize. They're going to attach to each other, and they're going to form strands with one wrapping around the other to form actin filaments or microfilaments. This is also microfilaments. Filaments. All right? So how about the building blocks here? The building blocks in the microtubules, they are called tubulins. These are tubulins. Okay? You have alpha tubulins and beta tubulins. You have two kinds. We have alpha tubulins and beta tubulins. That is the very reason why you see two shades of green. Okay? But I'm not going to bug you with the details. I did tell you that the intermediate filaments are less well understood, so I'm not going to bug you with the proteins that make up intermediate filaments. All right. So in eukaryotic organisms, some eukaryotic organisms can have a flagellum like the sperm cell. Okay? Female in human do not have any cell with a flagellum. Okay? Now, this flagella, one flagellum, several flagella. These flagella are going to be organized in a certain way. Now, in prokaryotic cells, we saw that the flagellum is made up of three parts. You have the filament that is made up of flagellins. These are proteins. We have the hook, and we have the basal body. Three parts, right? And we also said that the flagellum is powered by the proton motive force, right? Here, the structure is different. First and foremost, you have the membrane that covers groups of microtubules that make up the filament. So the filament is made up of groups of microtubules that are organized in a specific manner. How are they organized? The filament right here is organized in a such way that the microtubules are organized by two. We have 9 of them. So if we start here, we have 1, 2, 3, 4, 5, 6, 7, 8, 9 sets of 2 microtubules. Within the middle, 2 microtubules here. That is the very reason why we say that the flagellum of eukaryotic cell, the filament part, has a structural organization of 9 plus 2. Okay, 9 doublet and 2 microtubules in the middle. Okay, the motion of this filament, it's actually a whip-like motion as opposition to, well, not opposition, as compared to the prokaryotic flagellum that has a 360 movement. Okay, so the base here is different compared to the filament. The base, it's actually going to be made up of triplets of microtubules. And these triplets, you have 9 of them. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9. Notice that the middle has nothing. So here we say that the base of the flagellum, it's a 9 plus 0. Okay? So that's the organization. Now the membrane that covers the filament or the flagellum of eukaryotic cell covers the flagellum all the way. From the beginning, the base right here, to the very end. The plasma membrane covers it all. Okay? So the protists that are able to swim with a flagellum, that's the organization. The sperm cells, this is the organization. Okay? So beside the flagellum, eukaryotic cells are going to have other appendages, including the cilia. The cilia are going to have the exact same organization. The difference, however, between the cilia in eukaryotic cell and the flagellum is that the cilia are way shorter. But the part of the cilia that extend out are exactly organized just like the flagellum, and the base of the flagellum is exactly, the base of the cilia, is exactly organized the same way as the base of the flagellum. Okay? So cilia are going to allow the movement of some particles, either to attract some particles toward them, or to push some particles away. For instance, along your windpipe, you have a lot of cilia. Okay, these cilia are there to push the mucus that has trapped some particles that were in the air that you breathe. Some of these particles are bad for your health. As they get trapped, the cilia are going to vibrate in such a way that the mucus is going to move toward the throat, and when it reaches the throat, you're going to swallow that mucus that is going to go in your stomach, where the particles are going to go and get neutralized. Okay? The DNA, the nucleus. The nucleus is going to contain DNA. Okay? That nucleus, it's an envelope that has two membranes, right? It's a double membrane that is folded in such a way that you have it like this, right? Okay, so we have a folded membrane. Okay, so that's one membrane and the second membrane here. And you have here some nuclear pores. These nuclear pores are essential to allow chemicals in and outside of the cell. Okay? And this is very important because you have an exchange, a constant exchange of material between the cytoplasm right out here and the inside of the nucleus. For instance, DNA is in here, but RNA has to go outside. By the same token, we have the enzymes that are responsible for the synthesis of DNA that are produced out here. These enzymes have to end up inside of the nucleus so that the genetic information can be copied, right? So that's basically the role of the nuclear pores. Then we have the nucleolus. The nucleolus is this area that is very dense inside of the nucleus. That is where ribosomal RNA are synthesized and where the assembly starts. In fact, the assembly doesn't complete there, but that's where it starts. Then we have the mitochondria. The mitochondria are going to be very important to produce ATP. Now, I did tell you that the mitochondria have an outer membrane and an inner membrane. And the inner membrane is the place where the electron transport chain is going to be located. This is where the machinery responsible for the electrochemical gradient is going to be located. And because of that, you're going to have ATP production. Now, what, however, is interesting about mitochondria, it's that they have ribosomes. These ribosomes are 70S ribosomes, okay? Just like prokaryotes. In addition to that, mitochondria have DNA. And one can wonder, well, why do they have DNA? Well, that's a good question. And that's a question many scientists have asked, to the point that when looking at the structural organization and the function of mitochondria, it is thought that mitochondria used to live a life on their own, that they were actually bacteria that got engulfed and became, over a long period of time, interdependent with the cell that engulfed them. So the cell that engulfed them depends on mitochondria now, and the mitochondria now depend on the cell that engulf them. This is what we call the endosymbiotic theory. Okay? The endosymbiotic theory is the idea that mitochondria at one point in time lived a life on their own, but then got engulfed by a larger cell and they became interdependent. All right. Just similar to mitochondria, chloroplasts actually are also thought to have had a life of their own. Why? Because just like mitochondria, they have DNA, they have ribosomes that are 70S ribosomes. They are able to produce ATP via electron transport chain that are located in the membrane of the thylakoid right here. So this, guys, the chloroplasts are thought to have been some microorganisms, some prokaryotic cells that were photosynthetic. Okay, they evolved from cyanobacteria, and here we are also talking about the endosymbiotic theory, right? Then we're going to address the endoplasmic reticulum. In the endoplasmic reticulum, we're going to have the rough endoplasmic reticulum that has a whole bunch of ribosomes attached. Okay? This bunch of ribosomes are going to be there for the synthesis of proteins. Okay? Which means that the rough endoplasmic reticulum is associated with protein synthesis. On the other hand, you have the smooth endoplasmic reticulum. That smooth endoplasmic reticulum has no ribosomes attached to them. Okay? This smooth endoplasmic reticulum is going to be involved in lipid synthesis and lipid breakdown. Now, in some cells, the smooth endoplasmic reticulum is actually an area where calcium can be stored, for instance, in what do you call that? In muscle cells, right? You have some calcium that is stored in the smooth endoplasmic reticulum. Then we have the Golgi apparatus. The Golgi apparatus works alongside with the rough endoplasmic reticulum. Why? Because the proteins that are synthesized through the rough endoplasmic reticulum are going to end up in a vesicle. That vesicle eventually is going to travel to the Golgi apparatus, and in the Golgi apparatus, these proteins are going to be modified, they're going to be packaged, and after they are packaged, they are going to be sent to their final destination. And that final destination is basically three kinds. One possibility is that the modified protein is part of the lysosomes. Okay? When they are part of the lysosome, that means that these are digestive enzymes. Okay? That's one fate. Another fate is that these proteins are going to be integral part of the plasma membrane. So when the vesicle buds off from the Golgi apparatus after all modifications are made, that vesicle is going to travel, merge with the plasma membrane, and the proteins that are in that vesicle are going to be part of the plasma membrane. Okay, remember that the plasma membrane, it's a phospholipid bilayer with some proteins embedded. Okay, this is how those proteins end up in the plasma membrane. Finally, but not least, you have some proteins that are sent outside of the cell. I took you the example of what did we call it? Antibodies, okay, that are produced outside of the cell. All right. Okay, the lysosomes, they are going to contain digestive enzyme. I already told you how this enzyme end in the lysosomes. And this digestive enzyme break all kind of material, whether you're talking about carbohydrates, lipids, nucleic acids, proteins, you name it. They are able to break down viruses. They are able to break down bacteria, you name it. So they are going to break down about everything, not only particles that come from outside of the cell, but also particles that are very much part of the cell and that needs to be destroyed, like old organelles, right? We call this autophagy. Okay? The peroxisomes play a very important role in detoxification because they're going to allow the conversion of hydrogen peroxide into water and oxygen. That's an example of detoxification, right? The peroxisomes are going to be very important in breaking down lipids, okay, and other roles. And this is where we are going to stop with this chapter. Thank you for your attention. I will see you on Tuesday, February the 10th. Thank you. Bye now.